This week we are talking about RNA, and this is going to be a continuation of what we talked about last week on DNA, but there was just too much information to pack into 1 week, so we are moving on from there. RNA is required to make proteins from the DNA double helix. Proteins are required for nearly every activity and chemical reaction within the cell. We talked about proteins very early on in the course, and we looked at enzymes. Enzymes are a type of protein. So this process is vastly important. As you look at this process more in depth, remember these basic steps. So we're starting with DNA, and then we will move to creating RNA, and from the RNA we will create proteins. So first, let's talk about the structure of RNA. RNA stands for ribonucleic acid, and it's the other type of nucleic acid found in cells. It has another essential, although different, function from DNA. There are 3 differences between RNA and DNA. First, RNA is single-stranded. Whereas DNA was double-stranded in that double helix, RNA is just a single strand of bases. RNA has ribose sugar rather than deoxyribose sugar in its backbone. So deoxyribonucleic acid is DNA, ribonucleic acid, RNA. You should be able to remember ribose sugar in RNA, deoxyribose sugar in DNA. And finally, the 4 RNA bases, there are also 4 RNA bases, but this time they are adenine, guanine, cytosine, and uracil. Thymine is never found in RNA. So we'll keep working with that throughout our PowerPoint here. But keep in mind RNA has A's, G's, C's, and U's, never T's. So there you can see we have the sugar. This is the ribose sugar, phosphate backbone, and then you have your nucleotide bases, or nitrogen bases, from coming off of your ribose sugar, and you have cytosine, guanine, adenine, and uracil in a single-stranded RNA molecule. Okay, there are 3 types of RNA. Each has a different job in the cell. First, messenger RNA, or mRNA, carries copies of the DNA sequence outside of the nucleus. We call it messenger RNA. It's easy to remember. It's carrying a message outside the nucleus for the cell. Ribosomal RNA, rRNA, again, this one's going to be easy to remember. Along with proteins, these molecules make up ribosomes. And as we talked about when we were learning about organelles in cells, ribosomes are where proteins are made. Transfer RNA, or tRNA, is a clover-shaped molecule, you can see it here at the bottom, that matches an mRNA sequence to the amino acids that they code for. Amino acids are the building blocks of proteins, so all proteins are made of these tiny molecules called amino acids, and the tRNA is responsible for matching the sequence on the mRNA to the amino acid. And here's the amino acid that's then going to become the protein. We're going to talk way more in depth about all of these things. Each of these types of RNA will be further explained. They each have a job in the process of converting DNA into proteins, which is what we're discussing next. So our whole goal, remember from the first slide, is that we are turning DNA, the code from DNA, into proteins for the cell to use. So the first process in this sequence is transcription. Without DNA, the cell would be completely unable to perform its daily tasks. Therefore, DNA is well protected within the nucleus of the cell. So the DNA needs to remain in the nucleus, because if anything happens to it, there will be mutations, there will be errors, and all kinds of havoc will be wreaked on that cell. However, DNA is needed to code for proteins, which are made in the ribosomes outside of the nucleus. So we've got the ribosomes floating around out here in the cytoplasm and sometimes attached to the endoplasmic reticulum, but the DNA is located inside the nucleus and it needs to stay there. It cannot come out to the ribosomes and carry the code out because it needs to stay protected in there. So this is the little problem that we're going to start with. DNA is in the nucleus, but we've got to get it to the ribosomes somehow without affecting its code. Therefore, mRNA's job is to make a copy of the DNA sequence inside the nucleus and then take that copy out of the nucleus and out to the ribosomes. The process by which this copy is made is called transcription. So just like you'd think of a scribe or someone transcribing something like a medical code or something like that, a medical transcriptionist is responsible for writing down all the things that have been said during a doctor's appointment and codes and things like that. This is making a copy. Transcription is making a copy of something. In transcription, an enzyme called RNA polymerase, we talked about DNA polymerase in the last week, but this time RNA polymerase unwinds a small section of the DNA double helix, then uses one side of the double helix as a template to make an mRNA copy. So here we have our double helix in purple, and a little portion of that has unwound and unzipped so that the mRNA can come in here and make a copy of one side of the DNA. The template is easy to follow because of the base pairing rules. Normally we have A to T and G to C in the DNA, but whenever the DNA sequence contains an A, uracil is made rather than a T. So here in our copy, our mRNA copy, A is going to be bonded with U. C and G are still together. The thymine in the DNA still creates an adenine or bonds with an adenine. But you'll notice as you go down this mRNA molecule here, this strand, you'll see only A's, C's, G's, and U's. RNA knows where to start and stop the copying based on specific DNA sequences called promoters. So there's a specific code in here that will tell the RNA where to begin. Not all of the newly created mRNA sequence is required to code for the protein. So we've got some extra pieces in there. In order to reduce the effect of any mistakes in the transcription process, the mRNA sequence includes large sections of bases that are useless or nonsense. So essentially, if you're copying a huge amount of material, even though it seems sort of worthless, if half that material is nonsense, the chance of you getting a mistake in the half that's actually important is going to be a lot lower, because half the time you're going to be making mistakes in the nonsense portion. So this is one of the backup measures that a cell has to make sure that it cuts down on the number of mutations and mistakes in the sequence. So these sections that are nonsense are called introns and are removed prior to the next step in the process. The remaining essential portions of the mRNA sequence are called exons. The exons are combined to form the mature or finished mRNA sequence. So you can see here we've got an exon sequence, we have an intron sequence. They're not always the same length, but in this picture they happen to be. And we have another exon, another intron, and another exon. Those introns are going to be removed, spliced out of that mRNA, and then you have the completed, mature mRNA made from the exons. Okay, so as we continue through our process of transcription here, we're going to start talking about the sequence of mRNA that we have left. Just as letters in a sentence are combined into words, bases in an mRNA sequence can be read in sections. So if you have a sentence that's separated into words, that's how it's read. In an mRNA sequence, it's also separated into sections. those sections are called codons, and they are always 3 bases. So we don't, unlike a sentence where you have words of different lengths, this time we're reading specifically 3 bases at a time. Each of those 3 base codons codes for a single amino acid. Together, these amino acids make up the protein. On the following slide, you'll see how each codon signifies one of the 20 possible amino acids. All right, so here is a very complicated looking chart. It says at the bottom here, this wheel is read from the center out. For example, an mRNA codon of UAU will code for the amino acid tyrosine. So we're going to start in the middle of our chart here. This is the first base. It's either on the mRNA sequence, A, G, U, or C. So if our mRNA codon is UAU, we're going to start here with our U. And then the next letter in that codon is an A, so we're going to proceed out to the A. We have a choice of U, C, A, and G. We're going to U, A, and then the last letter is U. So that is going to code, that codon UAU is going to code for tyrosine, and that is the amino acid that matches with that mRNA codon. Notice from the previous chart that there are 3 stop codons. This signifies the end of an mRNA sequence for one gene. So let's go back and look at that. You have a stop codon here, you have a stop codon here, and you have a stop, these are 2. So we've got UAA, UAG, or UGA. Those are the 3 stop codons. Also, AUG is a special codon. If it is found after a stop codon, it signifies a start codon for the next gene. If it's found in the middle of a sequence, it codes for the amino acid methionine. So again, let's go back and look here. We've got AUG. AUG is a very important codon. You want to remember that. That's the only one that you really need to remember. AUG codes for methionine if it's in the middle of a sequence, or if it's starting right after a stop sequence, it's going to code for the start. AUG is the start codon. That's the only one. Any bases prior to a start codon or after a stop codon can be ignored. So let's take a look at the following mRNA strand for an example. And back to our picture with the double helix unzipping and creating that mRNA strand, let's pretend that this is the sequence of codes, that sequence of bases that's along that mRNA strand. UCAUG, CUU, GGC, UGAG. Okay, we are going to eliminate the U and C at the beginning of this sequence, and we are going to start with the start codon AUG. So anything that's in front of an AUG, you are going to skip. And we're going to divide that up then into a sequence of 3 letters, because that's what our codon is. So AUG, CUU, GGC, UGA, and then we've got 1 left over at the end, and we'll ignore that. And then we're going to go back to the circular chart and figure out what are those codons coding for. So the coordinating amino acid sequence would be start or methionine, which is the AUG. CUU is leucine. GGC is glycine. And then UGA is a stop codon. So this is our amino acid sequence. The amino acid sequences that your cells use are much, much, much longer than this, but we are going to use pretty simple ones just so you can see how it works. So practice using the chart to determine the amino acid sequence for the following mRNA sequence. Here's the sequence. So you can pause the video here, take a minute. You can either go back and look at that codon chart, or there should be a printable one for today's lesson, and you can use that. Remember to start at AUG. Okay, assuming that you are ready to move on and check your answer. Our coordinating amino acid sequence is start, valine, histidine, asparagine, tryptophan, stop. Because we are going to eliminate those first four bases and we are starting with AUG. So start, valine, histidine, asparagine, tryptophan, stop. If you did not get that answer, I would go back and take a look at that again. You will have more time to practice for your activity this week, but this is definitely an important skill for this week's unit. Okay, now we're going to move on to our second process, which is translation. So we talked about transcription, which is making mRNA from DNA. Translation, we've already kind of gotten into it, but this is making the amino acid sequence from the mRNA. So now that you know how the codons form the amino acid sequences, let's see how the cell accomplishes this. The process is known as translation. Translation is also known as protein synthesis, which makes sense since this is the process where proteins are made from mRNA. So I know that this is a ton of vocabulary, but a lot of this is really a little bit intuitive because you have translation. Anytime you're translating one language into another, you are changing, and we are talking about changing the mRNA code, translating that into a protein. Protein synthesis is another word for this process. Synthesis, we've already talked about, means making something, so here we're making proteins. This process occurs in the ribosomes. Again, we already addressed that. So here's our overview of the process of translation. First, mRNA carries the transcribed code from the nucleus to the cytoplasm and attaches to a ribosome. So it's coming out of the nucleus and attaching to a ribosome out in the cytoplasm. The ribosome aligns with the start codon on mRNA because, of course, that's where the sequence is going to start. tRNA molecules that match with each codon bring in the amino acid for that part of the sequence. So tRNA molecule, you can go back and look at the slide near the front of this presentation where we talked about the three types of RNA. tRNA has a side on it that has a matching set of bases to the mRNA codon, and then on the other side it has, on the other side of the tRNA molecule, it's carrying an amino acid that codon codes for. The ribosome moves to the next mRNA codon, and another tRNA brings in the next amino acid. The two amino acids bond together, creating the beginning of a polypeptide chain, which will be the finished protein. So the two amino acids bond together with what's called a peptide bond, and that whole peptide chain of amino acids is eventually going to be called a poly, which means many, polypeptide chain. This process continues. The ribosome moves, tRNA attaches, and new amino acids brought in, and then the peptide bond is formed between the amino acids until the stop codon is reached. This is a complicated process to put into words, so let's take a look at some images and visuals of this. So here we've got transcription. DNA is unwinding and making the mRNA. And then this part here is the translation process. The mRNA comes out into the cytoplasm. It finds a ribosome. And then these are the tRNA molecules. So tRNA molecule has an anti-codon on one side of it that matches exactly with the codon in the mRNA sequence. So if this is AUG, our start codon in mRNA. The anticodon is going to be UAC on this side of the tRNA molecule. That's how the correct tRNA knows to come in. And then here's the amino acid that's on the other side of the tRNA molecule. And this is what one individual tRNA looks like. This is the chain that has been created as more and more tRNA molecules come in and these amino acids bond together. The tRNA molecules leave, they are reused, but the chain of amino acids remains, and that becomes our polypeptide chain, which folds up to become a protein. Here is a different but slightly more complicated view. Here's our mRNA again. You can see the tRNA is bringing in the amino acids, these colored balls here. This is talking about the A site and the P site and all of that. We're not getting into all of those, all that terminology, but this is the ribosome, this green, whole green structure. It's made of two pieces called the large unit and the large subunit and the small subunit. But you can see the tRNA is coming in here, is dropping off the amino acid, it's attaching with a peptide bond, and then the tRNA is leaving. And then that ribosome will move down the chain, more tRNA comes in, and more amino acids are brought in until the chain hits the stop codon, and then it releases and folds into a protein. This process is best viewed, as I said, by video, so you can see the movement of each molecule. This video is a great summary. So I have the link right here, but since this is our last slide and the video does not play well as I'm recording the PowerPoint, I would stop the PowerPoint here and then go to the YouTube link that's in this lesson. And definitely watch this video. I think it will really put everything into context for you and you'll be able to see the whole process from start to finish. If you have any questions, feel free to contact Home Science Support and let us know.